In this video, we'll be reviewing a little bit about the history of drugs. And here are the learning outcomes that we'll be looking at throughout this video. Now, generally, when we use the term drug in today's society, what we are thinking about or what we are referring to is usually an illicit drug. But from a pharmacological point of view, the term drug is any molecule that will alter body function and therefore produce some sort of biological response. Now, this includes studying medicines, which are used for beneficial or therapeutic outcomes to cure, treat, diagnose, or prevent some such disease. But it also includes the study of molecules and drugs that can be harmful or toxic. And this could be a dose-related effect. Now, drugs can be used or referred to in many different forms. There is the chemical form, which is usually the long, unwieldy name that is used by chemists. Maybe pharmacists might use them, researchers will use them. A more useful way of referring to them is with their generic or the non-proprietary name. So there is a series of universally adopted official drug names. There is also a list which has been approved and adopted by the US, which is referred to the USAN, so US adopted names. There is also a British equivalent, and there is also an international equivalent, which has been designated by the World Health Organization. Then you will also find that drugs are referred to, or when they get released by a drug company, they will also have a brand name. And so there are a number of different ways that you can refer to the same drug. And we have a few examples here. So Panadol or Tylenol are trade names for what we would refer to in Australia as paracetamol. But if I was in the States, I would refer to this as acetaminophen. The chemical name is N-acetyl para-aminophenol, which doesn't roll off your tongue. And you can see from the other chemical names that they are not generally going to be used by patients. The trade names are generally ones that patients are more likely to get their tongues around and be able to pronounce. But when you're prescribing, the generic name is actually going to be the name that you'll use. Now, there can be some issues with using the brand name, although it's very pronounceable in the majority of cases. You can have problems with them. So you can have one drug with multiple names. So Febridol, Panamax, Paraglin, Parapain are all the same drug. They all are paracetamol formulations. And this complicates it when you're talking with patients because there are so many different ways of referring to the same drug. It also means there's more use of your memory to remember obscure names when there's really the only thing that's important is the active ingredient in the majority of cases. Sometimes there are differences in naming between countries. So the same company may produce drugs that are called something else when you go between countries. There are also the problems of looks alike, sounds alike names. So Derelan and Deptran, one is used for cardiovascular scenarios, one is used for depression. Arapax and Aprax, one's for depression, one's for pain. Celopram, Celebrex, one's for depression, one's for pain. And there are many examples where the wrong drug has been given to the wrong patient because it's been miscommunicated at some point in time. So from this year onwards, all prescribing is to be done with the generic or the active ingredients listed on the script. There's no use of brand names. Now for a little journey through history. So the use of drugs and medication has actually been going on for quite some time. So you can actually go and have a look at historical extracts and find uses of extracts of ephedra, cannabis, opium from the 1st century onwards, and it probably extends well beyond that as well. into the kind of BC kind of range. You'll see tinctures being used, which are alcoholic extracts. You'll see that nitrous oxide was eventually discovered, which is used for anesthetic purposes. Digitalis, or the source of digoxin for cardiac conditions, has been found and isolated and used as an extract. And then you can start to see that we start to get to synthetic substances in the 19th century or so. And you can see that where society has discovered that there are actually active ingredients within plant extracts. So morphine and codeine were derived from opium poppies, cocaine from coca, from the leaf, and quinine from bark. And each one of those was found to have its own purpose. And you didn't need the whole plant, you just needed the active ingredient. And from there, substances could be then synthesized without using the plant itself. So aspirin is a modified version of salicylic acid, which is found in willow bark. Narcotics have been synthesized, either semi-synthetically or fully synthetic versions have been developed, built on from the morphine and the codeine. And then you start to see the development of insulin, which is a human peptide. And eventually you see recombinant proteins replacing insulin derived from animals to insulin that's actually grown in a test tube from bacterial cells. Now, as far as drugs and where we get them from, there are many different ways that they can be derived. So I've already indicated where there is a natural source of medication or drugs. So plants, digoxin came from the digitalis plant, which is this kind of purple bellflower here. You can get conjugated estrogens, which were from an equine source, so from the horses. Microorganisms also produce a lot of useful drugs, so phenoxymethylpenicillin being the most notable of those. Also, things like minerals, iron, zinc, and selenium can also be sources of drugs. There are synthetic versions, so paracetamol and acetylsalicylic acid are synthetic. Semi-synthetics, paclitaxel, which is used for cancer chemotherapy, and that came from the yew tree and it's been developed from there. But you also have the biotechnology kind of side of things where the use of genetics and understanding of genetic programming allows insulin to be made without requiring a mammal to make it for them. Monoclonal antibodies is another example of the development of biotechnology. The other thing that we'll point out during this video is that pharmacology has many kind of divisions and subdivisions in it. So if we have a look in the center here, we've got pharmacology, we've got pharmacokinetics, which is exploring the way drugs behave when they're in the body and how the body interacts with those drugs, particularly looking at rates and absorption and distribution and metabolism and excretion is the field of pharmacokinetics. There is biochemical pharmacology where you're looking more at the nitty-gritty of how drugs interact with receptors and enzymes and the like. Then there's also systems-based pharmacology where you're looking at how drugs influence neurochemistry or cardiovascular systems or the gastrointestinal system, or how it modulates the immune system or the respiratory system. There are also other aspects that we can look at. So medicinal chemistry is a synthesis and exploring the creation of new drugs. Clinical pharmacology looks at the use of medication and the decision-making process around the use of medication. There's veterinary pharmacology because animals and humans have a lot of similarities, but there are also a lot of differences. So there is a field there. Pharmaceutical sciences can include things like looking at the formulation of how drugs are administered to the patient. Biopharmaceutics and biotechnology, so the use of recombinant proteins and monoclonal antibodies will fit into that kind of basket. Toxicology is looking at the toxic effects of drug usage or drug exposure. Pharmacogenetics is getting to a more kind of newer area of pharmacology. So we're looking here at the genetics of, well, how genetics alters drug responses. And then pharmacogenomics, which is a kind of offshoot, is now looking at how genetic variation affects drug usage. So actually coming up with the rules and the guidelines for saying, well, if this genetic variation is present, then we would recommend doing this instead of that. Pharmacoepidemiology is looking at the impacts of drug use for the treatment of diseases, but also looking at the adverse drug reaction profile of medication and its usage. And finally, the final one on the slide there is pharmacoeconomics, which is looking at the health impacts and the cost versus benefit of using various treatments and comparing treatment A versus treatment B, saying, is it worth the extra cost? And they look at things that, like how much does the medicine cost, but how much benefit does society gain from the use of this medication? So this just unpacks some of what we've already looked at. So pharmacology is really looking at the interaction of drugs with living systems at the molecular level, really so we understand how they work. Pharmacokinetics we've already talked about, which is the ADME, absorption, distribution, metabolism, and excretion. Pharmacodynamics is a little bit different, so that's looking at what the drug does to the body. So this is where the understanding the therapeutic outcomes and the impacts on homeostasis that drugs have. Pharmacotherapeutics is an exploration of the use of drugs, so which drug is better for the treatment of cardiovascular disease, or which one is better for diabetes. Pharmacogenetics is the study of genes which affect drug therapy, and then genomics is what to do about that. And finally, we'll look at the drug development pipeline. So basic research is required to even understand where to start the process. And this might uncover the pathophysiological processes and pathways and the receptors and enzymes that are involved in a disease state itself. Then once a target has been identified and verified that this could be a useful kind of target to look at, then lead compounds need to be developed. And so this will require the synthesis of the compound with an idea of its final use, i.e. that this drug has to be compatible with the body, needs to be metabolized. How is it going to be excreted? Will it actually be absorbed? And so those kind of things are also important in that drug discovery process. Then there's a preclinical phase where we're looking at safety testing. Cell and tissue and animal models might be used to test the lead compounds and to rule them in or rule them out. And this will also include pharmacokinetic testing to make sure that it's actually usable as a drug, not just does what it's meant to do, but actually gets to where it needs to go. Then we're looking at various phases of clinical trials themselves. So in phase 1 clinical trials, usually they're fairly small, maybe 100 people or less. And what they're exploring at this point is the population pharmacokinetics and adverse drug reaction profile. So how safe is it to use? What kind of dose might be in the acceptable and not toxic? Phase 2, they're starting to look at patients now. So there's 100 to 500 patients that might be exposed to the medication. And now they're starting to look at exactly what dose would be useful for that particular disease state that they're exploring. Is it a once a day dosing or is it a twice a day dosing? What kind of drug regimen is necessary to treat the disease, and which one's better and which one's worse? And also they'll start exploring and collecting data to find out what the short-term adverse drug reaction profile is as well. Then if that's successful, on to phase 3. Now we're looking at thousands of patients. It may be done at multiple centres, including at international centres. And so you're starting to get different ethnic responses to medication as well. And they will continue to monitor the safety and the efficacy of the drug as well. And if all of this works out, then it goes to the drug registrar of that country to be reviewed. So in Australia, that's the TGA. In America, that would be the FDA. So the registrar will review the benefits and risk of the medication to the public and make a decision on whether it's needed or useful for the public to have access to this drug before they register it as a medication. And then after it's onto the market, there will be phase 4 trials where they will continue to look at the medication. Is it safe? Is it effective? And so these are long-term data, which generally you can't collect. The other thing in the earlier phase 1 to 3 kind of trials, so you're starting to see larger and larger numbers of patients being treated, and also with different ethnic backgrounds, different levels and drug interactions and things like that will start to pop out, and particularly rare side effects and adverse drug reaction profiles. These kind of things will only be seen once you reach phase 4 trials. So back to the learning outcomes. So you should be able to define the term drug, which means we're not talking about illicit drugs, but we're talking about substances that have a biological effect. You should be able to recognize different ways of naming drugs, so generic versus chemical names and brand names. Appreciate the history of drug development, that this is not something that's recent, that drugs for the use of modifying body behavior has been going on for many centuries. Should be able to outline some of the current steps in drug development, looking at the various phases of clinical trials, what might be involved. Should be able to recognize that there are issues with using drug names and that there is potential for confusing one drug with another. And also you should be able to differentiate the fields of research associated with pharmacology.